Here's my biology project on how seed taste in pea plants evolved due to artificial selection. So the word evolution is used in biology to describe the change in the heritable characteristics of biological populations over successive generations. And today we know of many things that have evolved. Us as humans have evolved, other animals have evolved, and yes, even some plants have evolved. So today we're going to be looking at the Pisum sativum, which common name is field peas, and how it has evolved due to artificial selection. So the first sample of field peas were taken back in 1000 BCE, and we know that they were a key crop of the Neolithic agricultural revolution. So field peas evolved and now express two different traits, round and wrinkled. The round peas, which allele in the genotype is big R, codes for functional SBE1 proteins. And the wrinkled peas, which allele in the genotype is little r, codes for a non-functional SBE1 protein. So what this SBE1 protein does, it allows the enzyme amylopectin to arise, which is the functioning starch branch enzyme. So in the round peas, it expresses a higher amount of starch than it does in the wrinkled peas, because in the wrinkled peas, this enzyme, there is no amylopectin within it. So without the starch within the wrinkled peas, it allows for a higher water concentration to be absorbed, which allows for more sucrose to develop through photosynthesis, which evidently results in them becoming more sweeter to us and having more sugar in them, making them more desirable or more susceptible to artificial selection. So for this experiment, I came up with two hypotheses. The first one is due to artificial selection, varied initial allele frequencies within pea plant populations lead to fixation. And then the next hypothesis is, following artificial selection, wrinkled peas will have a higher fitness than round peas, which will increase allele frequency. So artificial selection refers to the selective breeding of an organism. When an allele becomes fixed, it means that it's the only allele that exists in the gene for that gene in all of the population. A fixed allele is homozygous for all members of the population, meaning that they have the same alleles in the genotype. And finally, the allele frequency refers to the relative frequency of an allele at a particular locus in a population. So now on to the methodology of this experiment. You can see in our figure 1 a good visual of the two phenotypic differences of the round and wrinkled peas, and it also shows the fields that the 10 plants of peas were grown in. And we started with 20 fields of peas for the whole experiment. And for the first part, we increased the initial wrinkled pea frequency by 0.05 every time to observe if the beginning frequency affected the amount of time it took until it becomes fixed. And then for the second part of this experiment, we observed the A1 allele, which in this case stood for the wrinkled pea phenotype, over generations with different fitness values. So we did this, and we changed the genotypic fitness values. A1A1 was 1, A1A2 was 0.95, and A2A2 was also 0.95. So in this case, the A1A1 allele, or A1A1 genotype, stands for wrinkled. And due to the fact that the wrinkled peas have the recessive allele, the A1A2 means round would result in round peas, and the A2A2 would also result in round peas. We started with a control, and we kept that fitness at 1 throughout the whole experiment. So moving down to our graphs, figure 2 is trend lines of the alleles that were more frequent in the beginning of the population and grew to be the most fit. So the blue line here stands for A1 allele, which is our wrinkled pea allele becoming fixed, and the orange line stands for the A1 becoming lost. So as you can see in the beginning, it was much more common for the A1 allele to become lost leaving no wrinkled peas in our population. So as we progressed and increased the frequency of the beginning by 0.05 each time, we can see that it gradually became more and more often that it was becoming fixed and less often that the allele was becoming lost. So by the end, or by 0.75, you can really see a big jump of the wrinkled pea allele becoming fixed way more often than it was becoming lost. And overall, the higher the allele frequency of A1 in the beginning of a population allows for a higher fitness in a shorter time before that allele becomes fixed. So now we're moving on to figure 3. And this shows the A1 allele, which stood for the wrinkled peas in every population becoming fixed after artificial selection. So in the control group, the first 2 bars, we can see that there was an equal time, pretty much equal time, of the A1 and A1 allele becoming fixed and the A1 becoming lost. In the next bar, we can really see that following artificial selection, the A1 allele became fixed 100% of the time. We can also see the variability of the control data was much lower in that group and then it was in the artificial selection group, and that is shown through our standard error bars. So overall, the pea populations that go through artificial selection result in the allele becoming fixed. So moving over to our results section as well, we have found that the larger the allele frequency in the beginning of a population results in the greater chance of that trait becoming fixed. It also results in a shorter amount of time until that allele becomes fixed. And that can be shown, like I said, in figure 2. And then after 10 populations of peas, we found that it displayed the wrinkled pea allele becoming fixed following artificial selection. And what this means is that the wrinkled peas, after going through artificial selection, will also have a result in becoming more fit. So we can also say that this is a significant difference between the alleles becoming fixed in the control group and becoming fixed in the after following artificial selection group. And this is because our t-test, which you can see here on the screen, proves that it's a very significant difference. So in conclusion, if we move down to our conclusion section, we have found that the more frequent the wrinkled pea allele in the beginning, the shorter amount of time until that allele becomes fixed, and the higher chance that it will become fixed. And we found that artificial selection, which I've said is the selective breeding of an organism, occurred at a higher rate with the wrinkled peas because they were sweeter and more desirable to us, and that the wrinkled peas have a greater fitness than round peas do after going through artificial selection. So that is how seed taste in pea plants evolved due to artificial selection. The more frequent the allele, the faster it was to become fixed. With that said, artificial selection led to wrinkled pea little r allele, resulting in a larger fitness, which affected the evolution of the Pisum sativum pea plants.